Okay, and I think we have everyone back. We are recording. And is there anyone who can't see my screen? Okay, good. Okay, so let's get back to the lecture. We were talking about different types of aligners. Quickly went through global aligners, which line from end to end, local aligners, which align short pieces of your query to your reference. And then there's local aligners, which we talked about some of the reasons that local aligners aren't necessarily the best for RNA-seq data. Local aligners being assuming that the full read is from one specific location to the reference. And the only way that that could work for RNA data is if you're aligning to or mapping to a reference genome. And so now we're diving a little bit deeper into the aligners, looking at the difference between splice-aware and splice-unaware aligners. And so as a reminder, splice-unaware aligners are not going to be able to properly align reads that span splice junctions, which is going to be an issue when we're trying to align cDNA or RNA-derived sequence data to a DNA reference. The only way that they could be used for RNA-seq data, again, is if you're aligning back to a transcriptome, and there's always caveats of aligning or mapping back to a transcriptome, depending on how good and comprehensive that transcriptome reference is that you're working with. Although there are some pseudo-aligners that do this, that do a pretty good job, especially for higher expressing transcripts. And so we had just ended with the definition of splice-aware aligners. So those aligners are equipped to handle intron size gaps, which of course improves alignment of reads that span splice junctions when you're aligning to a reference genome, which is why those are the ones that are commonly used for transcript-derived cDNA to DNA alignment. And then I just have a couple of different tools that are used to do these types of aligners. So some splice-unaware aligners that are commonly used. By no means is this list comprehensive, just some of the more common ones that are used are BWA. Many of you guys might have heard of this for splice-unaware aligners. There's also Bowtie, which is similar to BWA, and I have links to these in the PowerPoint so you guys can learn more about them. Pseudo-aligners, so two common ones that are used are Callisto and Salmon, and so these are the ones that use the De Bruijn graph to kind of map based on complexity of the read compared to complexity of the different regions of the transcriptome. You can read the documentation to learn a little bit more about how that pseudo alignment works, but I am also going to put a link in the chat here. There's this kind of GitHub post that goes through and explains how that type of K-mer-based comparison works, and I think that they do a really good job with pictures and stuff. So for those of you guys that are curious about pseudo aligners and how they work, I encourage you to check out that link. And the aligners that we use at GeneLab is a splice-aware aligner. So here's a couple of them listed here. The one we use is called Splice Transcripts Aligned to a Reference, more commonly referred to as its acronym STAR, with a link to that documentation. And then also HiSat2 is another commonly used splice-aware aligner, so I have a link to that tool as well. So as I mentioned before, we can use these tools to do the alignment. We of course have to have our reference genome file, and since we are using the splice-aware aligners, we will be able to align our reads back to a genome reference. And short read aligners, of course, require a reference. When aligning RNA-seq data, we use the reference genome of the organism in which the samples were derived. Don't see the splice-aware. So in the lower left corner here, there's splice-unaware aligners here. In the middle, at the bottom of the slide, there's pseudo-aligners. And then at the right here is where I have those splice-aware aligners. Okay, good. And so we're going to be using the STAR alignment, but we're first need to talk a little bit about the reference genome that we're going to need in order to align our reads back to, and of course we're going to choose a reference genome from the organism in which our samples were derived. Since we're working with samples that came from the soleus muscle of mice that were flown either on the International Space Station or kept on the ground for ground control, we'll be mapping or aligning back to the mouse reference genome. So these reference genomes are stored in FASTA files, which have this format here. So as was pointed out by Chief earlier today, the FASTA format is of course different than the FASTQ format that we have for sequence data. And so just want to break down this FASTA format a little bit. So we see that the sequence is here after this header line. We might see some long strings of N is highlighted in green here, and that could represent an assembly gap, like something where we just don't have clear sequence for. There's also could be these areas that are in lowercase letters, and that could mean because they're low confidence assembly or they're soft masked repetitive regions. So obviously, you know, our genomes have a lot of repeats throughout the genome, and so sometimes your reference will display those highly repetitive regions in lowercase. All FASTA files start with a header line that begins with a greater than symbol, as you see here. The second part of the header is an identifier or a sequence ID. So this could be a database specific ID, such as a chromosome number, which is shown here. It could be a gene symbol. It really depends on the database that you're pulling your FASTA file from. And then there's also these description fields. And so these description fields are just going to be additional information about the sequence. Some databases will maintain a standard format for this part of the header. Not all of them do. You'll notice here that the different sections are separated by spaces. Some will separate them by spaces, some will put a pipe in there, some will separate the fields by a comma. So here we see a couple different descriptions for this particular reference sequence. We also, in addition to knowing the actual sequence, we need to know the coordinates that are meaningful, or the ones that we know about that are meaningful. So those would be the ones that contain gene annotations. So if we want to identify those annotated genes, which are genes with known genomic coordinates and functions, we'll need to provide the alignment tool that we're using with a gene annotation file that corresponds to the reference genome file that we're using. So that corresponds to that FASTA file, but contains information about what is known about some specific regions on the genome. So gene annotation, there's several different types of formats of gene annotation files. So we're going to be using ones that are stored in gene feature format or gene transfer format, and those have this format here. And so this is an example of what you would find in a GTF file. I'm just going to go over each column here. So the first column is going to be the sequence name, so the name of the chromosome or the scaffold. And then I have what these are in brackets for this line that has the star in front of it. So this is the fourth line down on this example of this GTF file. So the sequence name here for the one that starred is going to be 3, so it's chromosome 3. The second column is going to be the source or the program that generated the GTF file. So here we see this came from Ensemble, and Ensemble used sequence from Havana. The next column is going to be the feature. So feature types such as gene, exon, CDS, start codon, etc. The one we have starred here is indicating a CDS region. The fourth column here is going to be the start location on the reference sequence. So this is where on chromosome 3, in our case, that this CDS feature starts. The fifth column is going to be the end location for that feature on the reference sequence. So for our example highlighted here, we see this end location is going to be here again with respect to the location on chromosome 3. Our 6 is a score. It's a floating point value. I think this one indicates a score based on how confident the annotation is, but I'm not 100% sure if one of the other instructors want to chime in. Numbers, the 7th column here is going to be if it's on the forward or the reverse strand of the DNA. Number 8 is a frame, so this indicates which base, 0, 1, or 2, is the first base of a codon. So basically any of these CDS features are codons, so it's going to tell you if this codon is starting at the 0 position, the 1 position, or the 2 position of that codon. And then starting column 9 here, these are where the attributes for this feature start. So it's basically a semicolon delimited list of tags with additional information. So in this highlighted one, we see here that it has information about the gene ID with the Ensemble ID format, which version of this annotation of the gene here, And then the respective transcript ID, again in Ensemble, is what's listed in this highlighted one here. Questions about GTF files or FASTA files? Okay. So where do we get these reference genome and annotation files from? Well, they can be downloaded from a variety of different publicly available databases. I'm going to list a bunch of them. So Ensemble is the one that we use at GeneLab. And then there's also Ensemble Genomes, which is part of Ensemble. There's GenCode, and GenCode actually uses Ensemble IDs. There's Illumina iGenomes, NCBI genomes. There's specialized databases that have annotated genomes, FASTA and GTF files, such as the flybase.org, wormbase.org, Fly-Drosophila, Worm C. elegans, ZenBase for Xenopus. There's VectorBase for looking at vectors, Phytozyme. There's Patrick BRC. I've actually never visited that one. So there's a variety of different places that you can get these FASTA and GTF files from. So again, at GeneLab, we pull all of our reference files from Ensemble, mostly because I really like how they document their versioning, how easy it is to grab files. Everything is very nice and neat and consistent across the board. So I'm a big fan of Ensemble. And this, we kind of went back and forth with our AWGs between Ensemble and NCBI. So those RefSeq references are also pretty commonly used. So as I mentioned, for these alignment tools, before we can actually start using them to align our trim reads, they need to index the genome. There's of course going to be a lot of information in the genome. So basically these tools need to create a genomic or transcriptomic index, and it allows the aligner to organize and store all that genomic content in order to make searching it a bit more efficient. And so that's the first thing that we're going to be doing before we actually align our trimmed reads, is build a genome. Now this only has to be done once for a specific read length for the STAR program. And so I already did this for you, so we'll just be going over it in the Jupyter Notebook. But just quickly, here are some of the options that are used. So STAR, you have to use a run mode parameter, and you tell it to do the run mode of genome generates. This is saying generate the index for the specific genome that I'm working with. And then there's this option here, the genome assay index n basis. So this value is relative to the size of your genome. It's basically just telling the program how to split up the genome to look at, I think, splice annotations in this one. And the really nice part about this is it tells you in the documentation what the calculation is that's used to determine what this value should be. So it's the minimum of either 14 or the log base 2 of your genome length divided by 2 minus 1. And so just as an example here, if you were to have a 1 megabase genome, the value would be 9. For mammals like mice and humans, we use 14 because those are larger genomes. But the really nice thing about this is you don't have to memorize this, you don't have to do this calculation. If you go and you generate your index for a reference genome and you put this value as something other than what it should be based on the size of that genome, the STAR tool will actually in the standard output tell you, hey, I think you could use a better value for this option, and it'll tell you which value to use, which is super convenient. We then have this option here that specifies the path to output the index directory. And then we have, a couple of these options that tell it where to find the FASTA file. So genome FASTA file is just going to direct it to where the FASTA file is that you want STAR to use to build the index. And then this option here specifies where the GTF file is to use. And of course, those GTF and FASTA files have to be corresponding. So you don't want to use a FASTA file from one database and a gene annotation file from another database. So for us, we use both our FASTA and our GTF files from Ensemble. And then this option down here is going to indicate the length of the genomic sequence around the annotated junction. So this is dependent on your read length. And so this should always be one less than the length of your reads. So basically an annotated junctions could be the full length of your read minus 1, right? So if it's, let's say, 150 base pairs and it spans a splice site, it could span a splice site such that 149 base pairs are on one exon and then one base pair is on another exon. So that's why this length is always going to be one less than your read length. And so once you build your star index, you can then run the star alignment. So Star is a remarkable program. It has a ton of functionality. You can really customize what you want to do with Star, and it can be very overwhelming if you've never seen these documentations before. And so I do have a link to the manual down here, which will tell you what all of these options mean. I'll go over them quickly now, but you do not have to memorize any of this. This is all, you go to the documentation and you can see what the option is. And for just about all of these options, there is a default value. So if you're really unsure what to use, you're very new to doing RNA-seq analysis, you can always start with the default values. And then as you learn more about it or read up on the documentation, you can start to fine tune these parameters based on your specific data. So this first option here called 2-pass mode, we actually tell it to map twice. And the reason for that is when it goes and it maps through the first time, it can identify unannotated splice junctions. And so you can identify novel isoforms of transcripts using this method. And so it'll go through and it'll look at everything compared with the gene annotation file, so the known parts of the genome that you're looking at. But then it'll also identify if a read spans a splice site that isn't in your gene annotation file. And so in the first pass, it'll identify where those are. And then you have it go back, now that you know where there's novel splice sites are, you can go back and have it align a second time. And this time, it will incorporate those novel locations into that reference index. So that way you can actually quantitate those novel splice sites as well. And so that's why at GeneLab we do it in this 2-pass mode, because we get additional information about unannotated isoforms by doing it this way. We have this option here, which just specifies where you're holding the index. And then this here specifies the type of output that you want. So this tells you to output your files in a SAM format, and to put anything that doesn't align, any read that doesn't align, just to add that at the end of that SAM file. And we're going to go over what a SAM file is. This outfilter type specifies the type of filtering. We use the by Sj out options, which has star spit out this tabular file to give us quantitative information about the splice junctions that it identifies. Again, so our users have splice junction information available on our repository for any data set, any RNA-seq data set. There's also this list of SAM attributes, so you can put things in your alignment file associated with each read that gives some information about the alignment of that read. And so it's encoded in these little 2-letter codes, and if you want to know more about the SAM tags, I have this link here where you can learn what all those different attributes are. The reason that we put them in there is because other downstream tools might be looking for specific tags to quickly identify information about your read and how it aligns. So for instance, if it aligned uniquely, it'll be a certain code there. If it aligns multiple locations, it might be a certain code there. So it just helps some downstream tools assess your BAM file and where your reads aligned a bit faster. And then we have these filter values that we can include. So outfilter multi-map nmax. This is going to specify the maximum number of loci the read is allowed to map to. So remember, our genome is very repetitive, as we've gone over a few times, and so especially for those reads that are shorter, so we gave it a 20 base pair cutoff, right? So the shortest read that we have in any of our samples is going to be 20 base pairs. But of course, if it's 20 base pairs, that could, especially on the lower end, map to multiple locations on the genome. And so here you can say, you know, we'll keep it so long as it maps to fewer than the value indicated for this parameter. So we use the default value of 20. So basically any read that maps to more than 20 locations on the genome, we don't align it. We consider it an unaligned read. There's this outfilter mismatch nmax. This is the maximum number of allowed mismatches to be included in the alignment output, and we use a default value of 999, mostly because we focus on this next value here. So this could be based on your insert size, right? So the reason we use 999 is if we have like a really large insert, we could allow it to have up to 999 mismatches based on the reference genome. as long as it meets the value for this next option here. So this value, this outfilter, mismatch, and overread LMAX, this is a ratio of mismatches to the read length allowed for something to be considered a line. So at GeneLab, we set this value to 0.04. So that'll indicate that we will allow 4 mismatches for every 100 bases. So if a read aligns, if you have a 100 base pair read that's aligning to your reference, it can handle up to 4 mismatches in a location and still be considered that it maps to that location. And then this next value here, align intron nmap, this is a minimum intron size. A gene gap is considered an intron if its length is equal to or greater than this value. So this is basically saying what's the minimum size that you'll consider an intron. And so we call this 20. So basically if it's less than 20, it'll be considered a deletion as opposed to looking for, so if you have a read and it aligns, but then up to 20 base pairs don't align, then we'll say, okay, that could be a gap. It could be a deletion that happened in your sample of interest. But if it's more than 20, then we'll assume that it's probably spanning an intron. And so it'll go and it'll look for what could be another exon that it's aligning to, that part of the read is aligning to. Even more parameters, again, a lot of things you could do with STAR. So we have this align intron max parameter, which is the maximum intron size that you allow. So we allow really large introns because there can be really large intronic regions. So we use a default value of 1 million. There's this align mates gap max, and that's the maximum genomic distance between 2 mates of paired end reads. So this is, remember, for paired end sequencing, we have forward and reverse. And so we're saying how far apart can we allow that forward and reverse read to be, or basically how long of an insert are you okay saying that you have. And of course, this is going to be based on your filtering. We use the default value here of 1 million. Of course, how we filter, we're probably not going to get a 1 million long intron, which is fine. That's just the maximum cutoff. Then we have this align Sj Overhang Min. This is the minimum overhang allowed for unannotated splice elements. So basically, if you have a read, it's saying you how many base pairs of that read will you consider to be a splice, to be part of a splice junction that's unannotated, so an unknown splice junction. So basically, if it aligns, if you have 100 base pair read, and it aligns 92 reads to one location on your reference genome, and then there's an intron size gap, it wants at least another 8 base pairs of that read to align. So the last 8 base pairs of that read to align to another place on the genome if it's an unannotated splice junction, like a splice junction that we don't know exists yet. So this is a bit more stringent than this next parameter here. And this parameter is the minimum overhang for annotated splice junctions. And so we allow it to be 1 because annotated, we know what those splice junctions are. So even if it maps 99, the 100 base pair read, 99 base pairs map to exon 1, and then as long as that last base pair maps to exon 2, then we're confident that it spanned that splice junction, so long as exon 1 and exon 2 are known exons in a known isoform of that transcript. We have this Sjdb Score, which is an additional alignment score for alignments across database junctions. So this is just a score relative to how confident you are in the alignment across a junction. The SAM type here specifies the format. So we use BAM sorted by coordinate, which basically tells us to output that SAM file as a compressed version of that file, because SAM files are huge. So the compressed version of a SAM file is a BAM file. And then sorted by coordinate means that we want the alignments inside that BAM file sorted by coordinate. Quant mode will indicate the quantification desired. So we use transcriptome SAM option, which instructs STAR to output a separate BAM SAM file containing alignments to the transcriptome, which means that when we run STAR, we get 2 BAM files. We get one aligned to genomic coordinates, and then we get another one aligned to transcriptomic coordinates. We have this other option here called outsam header HD, and that just indicates that there's going to be a header line in the BAM SAM file, and the header line in a SAM file is just an indication of all the features that it can expect to find in that SAM file. An out file name prefix, so we could tell it what prefix we want to have on all of our output files. So we use the sample ID or the sample name for our prefix at GeneLab. And then finally, this last option here, read files in, is just pointing to the location of the trimmed reads that you're going to align. I know that was a lot. I did want to go over it so that you at least heard all of these things once. But again, you don't have to memorize any of this. It is all in the STAR manual. And so if you're ever unsure about what an option does, you can go and look at the STAR manual and it will tell you what that option does. Any questions about alignment before I go over alignment outputs?
Amanda, who developed STAR? Can you talk a little bit about that and who's working on it and if it's being updated?
This guy, Alex Dobin, it's actually, if you go to his GitHub, that's where STAR is. So with this link down here, oops, you see it's the GitHub for Alex Dobin. I don't know where he originated from, but yeah, he developed this and he's the main person that maintains it and updates it regularly. And he's really good about, like, remarkably, because a lot of people use STAR, about answering questions and like users, if they identify issues with things or if something's unclear, you can message. There's all sorts of different forms of BioStar and other, I don't know if maybe some of our, yeah, I think so, but yeah. So Cold Spring Harbor is where he's at. Cold Spring Harbor Laboratory. It's in Massachusetts. No, that's in New York. Cold Spring Harbor.
It's in Long Island.
Yeah.
It's
So
where the original eugenics archive started, so they're doing a lot of penance now because they
oh
were big
yeah.
time, bought in, as did most of the country, into the eugenics movement early in the century, last century.
Yeah, absolutely.
So I just always think about the future of these things we invest in. Is that a concern? How are, thinking maybe this is not the time to talk about how we maintain these resources that are really important for the whole community, and one person is doing all the sort of heavy lifting.
Yeah, so although that person does a lot of the heavy lifting, like it's such just developers in general are so good about building just a really collaborative and helpful community. So people will message, Alex and say, "Hey, I think you should add this feature to the tool system." And a lot of them will provide code. I wrote this code that I think would fit well into your program to perform this function that STAR doesn't currently perform. And then, Alex and his team will go through and look at those suggestions and try them out and test them and then put them in new releases of the tool versions. But it is a concern. And one of the reasons why at GeneLab, we revisit all of our pipelines every year because new tools and new functionalities are coming out all the time in bioinformatics. It is just such a fast moving field. Like, for instance, the first time I did anything with bioinformatics stuff was about midway through my postdoc. I was doing an RNA-seq study, and back then we were using, that was only 5 or 6 years ago, and we were doing Top Hat was considered the
Yeah, I remember Top Hat. Yeah.
and it uses, I think, bow tie underneath the hood to do the
Yeah,
alignment.
it does. This is bow tie.
So
Yeah.
it uses a global aligner, which we know is not good for RNA-seq data. And a lot of people still use that tool. And even the authors are like, guys, stop using this tool for RNA-seq data. There's better tools out there that does a much better job handling RNA-seq data. And so that was what, 5, 6 years ago, that was the gold standard still. And so that's how fast then people like picked up and started using STAR more and more, realized, oh, this is much better, more accurate. And so it could change. And yeah, that is a concern. And that's why we revisit these annually. That's also why we have our analysis working group members just as part of the community, because they may identify and pick up on new or better tools or even different options for these tools. And bring them to our attention or say, hey, I think I should update this for this reason. And so yeah, we're constantly looking and making sure at least annually we're revisiting everything and updating accordingly. Also the genomes too, the reference genomes. There's new versions of the reference genome that come out every so often, and there's also tweaks to the current version that come out. And so as we learn more about our reference genomes and as more information comes out, those are updated too. And so that's another thing that we update annually as well.
So how much does industry contribute to this? I mean, we have a lot of uncompensated labor in academia and science who work really hard for really low salaries. And so, and then we have all the private sector that the CEO is earning 30 million or billion and the worker earns like 20,000. So do they contribute as far as you know? Or
I
can you?
don't know the answer to that question about how much the private sector contributes to these kind of standard tools. Does anyone else on the call have any idea of that?
I started typing, but I'll just talk. Most paid software that I'm aware of, note that I'm not very aware of a whole lot of those tools, often just wrap open source software in interfaces that work for people who aren't familiar with the command line. There are some exceptions, um, but that's been my experience.
Yeah, that's a very good point. There are even like Illumina has a genome space, which I'm sure you might have heard advertised at the end of their sequencing by synthesis video, and they have like wrapped pipelines to process RNA-seq data that you can pay for. You pay for a subscription to genome space and can increase your storage amount of data that you can hold there and stuff by paying more, and they're using Star under the hood. And so there are, yeah, as Rachel mentioned, a lot of
So they're the Amazon of sequencing.
yeah, for sure. At least short read sequencing.
Thank you, Amanda.
Yeah, no problem. Yeah, so a lot of companies will take advantage of the work that people did in public sector, like at universities and stuff like that, and make money off of it. Yay,
There
capitalism.
are a few tools that people use that are proprietary and that they are paid, but oftentimes those aren't open source. And so when I see those as a reviewer, I'm always somewhat skeptical because it's hard to know what's going on under the hood. I think that the one I'm thinking of is CLC Workbench.
Mm-hmm.
It's a good point, Rachel.
Yeah, that is a good point. And yeah, so even that's true, even some of the private sector tools that come out that might be publicly available, won't tell you, right? It'll be proprietary. So you can't like play with things or manipulate them. So because GeneLab's an open source project, we really try to stay away from anything that has anything proprietary or a cost associated with it, just to try to stay as transparent as possible and to have more control over what's happening under the hood. So, for instance, most recently, Lauren, one of our co-instructors, is working on a single cell RNA-seq pipeline, and originally was thinking about using Cell Ranger, which is a platform that uses 10x technology, and a lot of single cell data right now coming off uses 10x technology, but there are other types of companies to generate single cell sequencing data. And so because Cell Ranger is their platform, which you can use for free, you can download and install it for free, but what goes on under the hood is proprietary, and it's pretty rigid. And so Star actually, someone approached them several years ago and was like, hey, you should make a version of Star for single cell data. And they actually tried to deconstruct the Cell Ranger algorithm, and they have a version of Star called Star Solo, where they try to mimic to get the same output as Cell Ranger does, but this time all of the options and stuff are, shown, available, and usable. So Lauren's been working on doing a comparative analysis of Cell Ranger and Star Solo. That's been trying to go with Star Solo because of the transparency aspect of it. Yeah, good questions. Other question? So Julie said in your field, the ones that are proprietary have neural networks. Yeah, in essence, black boxes that cost $70,000 to license. Yeah, those black boxes.
I need my frown reaction button, but I don't see. Well, there it is. Not really. It's not. It's sad. I'm not sad. It's another. It's one missing there. Yeah. I mean, I think this is so unfair, and it just adds to the divide between the first world and the developing world. It just creates greater and greater and greater inequities.
Yeah, for sure. Any other questions about alignment before we move on to what some of these outputs look like? Okay. So one of the outputs from the star alignment tool is a log file that kind of gives a summary of what happened in the alignment process for your trimmed reads. And so a couple of things I'll point out here. There's obviously a lot of information here. Again, this is all very well documented in the star documents. But some things I want to point out here is you'll see that we have a line here for the uniquely mapped reads. So just above this is the total number of uniquely mapped reads. And then it does it breakdown based on percentage of total reads. And so you see way at the top here, number of input reads. This is the total number of reads in this examples that were assessed. It tells you, what the average read length is. Then it tells you information about the alignment. So this being the raw total number of reads that aligned uniquely, so mapped to just one location on the reference genome. And for this example, it was 66% of the reads. And then it gives you some more information like the average mapping length. It gives you information about the number of splices that it identified, the number of annotated splice sites. It also gives you the number of non-canonical or unannotated splice sites. Again, if you do the 2-pass mapping, it can do this and do the quantitation of things that aren't even in the GTF file yet. So again, you have the ability to identify novel isoforms, which is pretty cool. It gives you some information about like the mismatch rate. So remember, we allow a 0.04% mismatch rate. It gives you a deletion rate per base. A bunch of information. I'll point out some other important things. So number of reads mapped to multiple loci. So again, a raw number here for the total number of reads that map to more than one location. And then that same number break down in percent of total reads. So we see here we had 66% uniquely mapped, 20% was multi-mapped, but like 86% of reads mapping in total, which is good. And you'll see it that high for well annotated genomes. We also see here, down here, you can see the number of unmapped reads that didn't map because they were too short. And so remember, we said that we needed at least 20 base pairs to match up in order to be counted. And then we have this one here, I guess it's 0, that's why I didn't point out, but just above it is the number of unmapped reads that had too many mismatches. So remember, we gave it a 20 mismatch threshold as well. So a 20, if it mapped to more than 20 locations, it would be considered unmapped. And if there were any reads that did map to more than 20 locations, it would be here. This, we also gave it a value in order to map to reads. A certain number of those base pairs had to map in order for us to consider it, and we used the default value of 20 there. And because of that 0.04%, if we had like 100, or if we had like a 20, what is that, 4% on that, so 1. Yeah, so this is like, we have 21 base pairs or something, all 20 of those, 20 out of the 21 for a read length of 21 would have to map in order for it to meet the short cutoff length for being too short of a read in terms of how many base pairs align. Then we have information about chimeric reads too. So a lot of information in the star alignment log. We are going to generate this with our data, and I'm going to prompt you to ask questions. I'm going to ask you questions to get you thinking about the information in this log and what it means for the samples and how well those inserts aligned to the reference. The other output that we get is an alignment file, right? This is our main file that we get here. It's in SAM format. Really, ours is in BAM format because of the option. We tell it to compress the SAM file. And so SAM stands for Sequence Alignment Map. B is binary. So this is a binary alignment map if it's in the BAM format. So again, just a compressed SAM. And so these SAM files have the following format. So I'm going to go through each line again. We're also going to take a look at this in our data, and I'm going to ask you questions. So try to keep in mind, and we can refer back to this slide here for what each field in this SAM file tells you. So the first line should look familiar. Again, I'm doing the same thing here. So the column number, what that is, a description of what it is, and then in brackets here is going to correspond with what that value is for the field for the line that's starred. So here, this is going to be everything for the second starred line. And so this first field should look familiar. This is a QName or a query template name information. Anyone know where this information came from? Guesses here? It tells you in the description, but Where did this information for this first column come from? Should look familiar. The sequencer, yes. Specifically, that first line of the read. Yeah, trim FASTQ file sequence name. Well, it's the read information. Yeah, so information that's found on that first line of the FASTQ file. If you guys remember, this is information about the flow cell that was used, the sequencer, the cluster location. It's essentially like that address of where this insert came from. And so again, keep in mind that this is going to be the same for the same insert of the sequence. So both forward and reverse reads will have this same Q name here, as we saw when we took a look at our forward and reverse read files for our trim data. The other thing is if you see multiple duplications of this, keep in mind that this is an alignment file, so it's telling me everywhere each read, or each insert I should say, mapped on the reference genome. And so if it was mapped to multiple locations, you're going to see a same read appear multiple times for every time and mapped to a different location on the genome. Keep that in mind, it's going to come up in the questions. Our next column here is a Bitwise Flag, and it's just information about the alignment that's kind of encoded in this numeric code here. It's really nice if you want to decode the SAM flag. The Broad Institute created a publicly available SAM flag decoder, which link is here. So we can we see that this is 83. So let me just go to this location here. So this super convenient, tells you information if it's a paired read, mapped in a proper paired, if it's a read mate, reverse strand, mate strand. So that's what the information is encoded in here. So the one that we were looking at is 83. You just type it into this nifty little tool and click explain, and it'll tell you it's part of a paired read. It's mapped in a proper pair. It's the read, the reverse read. It's the first in the pair. Or reverse read strand, sorry, that's where it maps to. And this is the first read in a pair of reads. So again, that's just a little code to tell you information about that read and how it aligned. And you don't have to memorize them. You could just go to that nice tool that the Broad Institute wrote if you want more information about it. Number 3 is the reference sequence name. So this is going to be the name of the reference sequence that the read aligned to. Of course, we used Ensemble FASTA and GTF files, so our reference is all in Ensemble IDs. And this is looking, actually, remember I told you that we print out 2 SAM files, so we actually print out a SAM file that indicates mapping to the genome, and we also print out a SAM file that indicates mapping to the transcriptome, so it looks at transcriptomic coordinates. And the transcriptomic coordinates are the one that we're going to use downstream. So this is the one that we're looking at now. And so this is an Ensemble transcript ID is what we're looking at. And the one for our highlighted star is here. It ends in 31314. Again, that's referencing an Ensemble transcript ID. Our next column here is going to be the one base leftmost mapping position. So the position on the reference genome in which the leftmost base of the read aligns. So if you have your read 503.n, the leftmost base in which it aligns or maps back to your reference genome is going to be this value. And then this map Q is a mapping quality. So it's equal to the negative 10 log, base 10 log of the probability that the mapping position is wrong. 255 indicates mapping quality is not available. That means that it's uniquely mapped. So basically this value corresponds to if it's a multi-mapped read, what's the likelihood or what's the quality of it mapping to this specific location. So you'll see here that this read we see is mapping to multiple locations. You'll notice there's different transcript IDs here in the same exact read header, right? So this read, and this read is all the forward and or reverse of the same insert because it all has the same read header. But you'll notice that it's mapping to 2 different transcripts here, right? So this, in my mind, our HCs are 2 of the same. So this is probably a forward and a reverse. of an insert, and this is also a forward and reverse of that same insert, mapping to two different locations. And so this quality score is basically saying how confident are we that it maps here versus here, and you can see the quality for these are the same. And that has to do with, remember, we allow for a certain number of base pair mismatches, 4% to be exact, base pair mismatches between the read and the reference. And so this quality can come, right? So let's say if your read maps to one location and it maps exactly, then it's not going to have any mismatches. But if that read maps within a 4% to another location as well, that will also come up as it mapping there. And so the mapping quality score is going to be better for where all of the base pairs of the read maps versus to where there might be a few mismatches. So that's what that quality score indicates. And 255 is, again, a uniquely mapped read. We have this other thing here, which is the cigar string. So it's the aligned read length and the associated operation. So this encodes information about the alignment relative to the reference. It'll tell you if it's a match or a mismatch, insertion or deletion based on the letters here. And you can read more about that within the documentation for the SAM format. Link is down there. That's also linked in the Jupyter notebooks as well. This 123, that is the insert length, and then M means that it matches. So it's a match of 123 base pairs. So it's telling you how many base pairs are matching that location on the transcript, that transcript location. The next column here is the reference name of the mate or next read, reference sequence name of the next aligned read in the template. So as it's going through the reference template, the next read that it hits, if it's the same, it's going to be represented with an equal sign. So basically, your mates that are next to each other, those will be equal signs. So we have mates here, equal signs, mates here, equal signs, and mates there. Now, the 8th position is going to be the position of the mate or the next read. So the one base position of the next aligned read in the template. And so this, right, so here's the leftmost aligned read of this one, and this is telling me that the next aligned read actually aligns at the same location as this one. So that tells me that that's forward and reverse read that's overlapping probably. And then number 9 here is the template length. So this is the observed template length from the leftmost position of read 1 through the rightmost position of read 2 that tells you the insert length that it sequenced in, again, the forward and the reverse direction. And so for this one, that read length is 123 base pairs. And then the sign that's indicated is if it's mapping in the same orientation of your reference or in the opposite orientation of your reference. And so we see here that, so this is another indication that these are mate pairs, is that one is in one direction relative to the reference, and then the other one is in the other direction relative to the reference. You see the sign differences with the insert lengths being the same. And then this, I shorten this because this is the actual sequence in your read. So this is your read sequence, the same thing that you would find in line 2 of your FASTQ file. And then column 11 here is going to be the ACA2, the Fared score, to indicate the quality of the respective base. So again, this is going to be line 4 of your FASTQ file. That information is there. And then the additional SAM headers. So I told you for each read, we could add these headers, and those are 2-letter codes that mean things about like how it mapped, where it mapped, the quality of the mapping. And again, those are used for some downstream tools for shortcuts to identify information about your aligned read. And you indicate that with the OutSAM attributes option that I talked about quickly in the different options for this tool. Questions about SAM files or the information in it? Okay. You guys are going to be looking at these in our data, and you're going to be asking questions. So I'm going to have you interpret what it means in these columns in the hands-on portion. Okay, so a couple more slides before we're going to hop back to the Jupyter Notebook and actually align our data. One, just to let you guys know, if you were to want to take a closer look at how your reads map to your reference genome, there are several tools available that can help you take a look at and view your SAM files. This is just links to a few of them. So there's IGV, there's BAMView, IGB BioVis version, there's Genome View, SAMscope, UCSC Genome Browser. And I just have, this is an image taken from UCSC Genome Browser. So this is looking at an alignment file. And so this just kind of gives you a visual representation of where your reads align to with respect to your reference. And so if you ever want to visualize where things are aligning to in your reference for each of your samples, you can take that BAM file and view it in like one of these genomic viewers. So now that we have aligned reads, what do we do? Well, a lot of information can be derived from alignment data, such as differential gene expression, which is what we're going to be doing. But of course, before we do that, we would first have to count those aligned reads. We'd have to quantitate them in order to compare expression between two groups of samples. We can identify novel transcripts, right? So if that's something you're interested in, you ran your alignment in the 2-pass mode, you could take a look at those non-canonical splice sites, and if you quantitated it, you can look more deeply at the genomic coordinates, right? Because all these BAM files have the genomic coordinates of where these reads align to. So you can identify novel transcripts, looking at those novel splice sites. Actually, this is just novel transcripts in general, so ones that aren't annotated at all. And then this next bullet point is specifically looking at known transcripts that might have identified a novel splice isoform, so a different combination of the exons that hasn't been annotated before. We can also look and identify variant calls, so you can identify single nucleotide differences between your samples and the reference. You can also identify indels, insertions, and deletions in your samples relative to the reference. You can identify and possibly quantify, depending on how deeply you sequence, things like long coding. Again, this is only if you did ribodeplete, but things like long known coding RNAs, pseudogenes, small RNAs, small RNAs, I just listed a few of different types of small RNAs here. Again, that also depends on your filtering and your cutoffs that you used previously. You can identify there. So a lot of different things that you could do with BAM files once you have them. So super useful. We're going to be focusing on differential gene expression though in this bootcamp, as you know. And so now that we've talked about alignment, we are going to align our data. So I want everyone to jump back to their Jupyter notebooks. And once you're back here, make sure that you're seeing the Python Conda environment GL4U idle down there in the lower left. Is there anyone who's not seeing that? Okay, good. Okay, so we have about 17 minutes before lunch. We'll see how far we get into the notebook. So as I mentioned, we left off after we checked the quality of our trim data. So we have trimmed FASTQ files now that we checked the quality of. You guys all told me that the quality looks good. We don't have adapters anymore. We're ready to align these. So as I mentioned in the lecture, before we can align them, we first have to build a start index for the reference genome using the reference files. And that's in order to be able to identify which gene each of those RNA sequences came from. And so as I told you guys in the lecture, we're going to be using the star aligner to do this. And since our samples came from the soleus muscle of mice, we're going to align the trim reads to the mus musculus reference genome. And this is the version of the genome that we're using, grcm38.p6. And that file is going to be a FASTA file that we got from the Ensemble Database found here. So if you guys just were to click on that link, this brings us to the Ensemble Database, and you'll see there's a ton. If you just wanted to look at like one chromosome, you could just align back to one chromosome, let's say. There's a variety of different types of genomes here. If you do top level, it actually looks at different haplotypes, which confuses STAR a little bit, so we don't use that. What we're going to use actually is this file here, which is the primary assembly for the whole genome. And so that's the one that we use. And it looks like this, right? You saw as I highlighted there. So we just download that. And then in addition to the reference genome, we're also going to need that gene annotation file that tells us all the gene features of at least annotated genes in the reference genome. And then just as a reminder, the annotated genes are ones whose genomic coordinates and often function are also known. And so all the gene feature information star needs is going to be provided in the GTF file, again from Ensemble. So just a link to where it is, and it needs to correspond to the same genome version, the FASTA file that you use, the same version of the genome. And so just looking, here's where that link goes to. So we use, and again, you could do it based on chromosomes, but we're going to use the full GTF file, which is the bottom line here. So Mus Maculus, again, you see the same GRCM38. This is release 101. So anytime there's slight changes made in the Ensemble version of the references, it's identified by different releases. So you want to use the same release of your annotation file as you do your FASTA file. So we're currently working with release 101. It's not the most current release. So releases happen every like month or two, sometimes 3. You'll see new updates added. And so again, we update our pipelines annually. So last we updated this, it was 101. And if you guys want more information about the GTF file, I know I did just go over it quickly in the lecture, but if you click this link here, it'll just give you more information about GTF, and corresponding of that is the GFF file. So before we aligned our trim reads to the reference genome, we're going to need that STAR program to organize all that information that's provided in both the genome and respective GTF files in order to be able to accurately assign each read to the gene from which it came from, and to do it in an efficient manner. And so again, this is the same command that I went over in the lecture. So we'll use STAR in the run mode called genome generate to tell it that we're just generating a reference index. And this you only have to do once per read length. And if you guys remember, we have a mix of read lengths in our data. And so you always want to go with the largest read length that you have for this overhang. Because again, it's saying up to how many can we have match with one base pair going to another location on the reference genome. So again, if it spans a splice site, let's say, and since we have 150 base pairs as our longest read, we want to set this to one less than that at 149. And then I went over all of these options in the lecture, so I won't do that to you guys again, but just so you know, that information is also here in the parameter definitions on the Jupyter Notebook. So I have this information in 2 locations, both here and in the lecture material. And for this command to generate the STAR index, you need a FASTA and a GTF file, are the 2 files that you need as input. And then your output data is going to be a STAR indexed reference, and it consists of all of these files. So again, this is just a breakdown of the information that's in that FASTA and respective GTF file. And it breaks it down in different categories. So looking at chromosome length, chromosome names, start sites, exon sites, gene sites, splice annotation information. So it kind of breaks it down into this format so that STAR, when you're doing the alignment, can easily find information based on the coordinates where it's going to align to. Again, only have to do this once. So I already did it for you so we can all use the same reference since we're all using the same data with the same max read length. So I did just want to go over that. Any questions about generating a STAR index or why it's needed? Okay, so then once we have that STAR index ready to go, we're then ready to align our trimmed reads to the reference genome. And so this step of determining the genomic location and ultimately the gene from which each short sequence was derived is a pretty challenging thing to do, as we went over in the lecture. Proud of you guys to think about any idea why that is. I know I have the answers here, but if you guys just want to put it in the chat, like why, some reasons why it's difficult to try to figure out where these sequences came from with respect to the reference. The answers are all right there. If you guys want to unmute your mic and just, or type in the chat why it would be challenging to identify where these short sequences came from.
it can contain a lot of unique, non-unique sequences,
right?
which
Yeah.
can make mapping difficult. Yeah.
Yeah, that's a good one. So it can map to it could be from different locations. Um, some other things is we're dealing with really short sequences in a really long genome. So reads are going to be very short relative to the transcripts where they came. And some genomes can be quite large. And as you just mentioned, contain a lot of non-unique sequences, making that alignment difficult. They have to cope with, right? So this reference genome is a compilation of genomes from various organisms, of course, with heavily inbred model organisms like we have with mice. There shouldn't be as many differences, but there are going to be some differences, right? There are individual differences from one individual to the next, even with some are highly inbred model organisms. So the liners are going to have to be able to deal with mismatches in the trimmed sequence data that you're trying to do and how it maps to the reference genome. There could be mismatches there. There could be insertions and deletions. It has to deal with that. Many organisms have introns in their genes, and therefore reads align non-contiguously, so not necessarily touching. So placing splice reads across introns and determining exon-intron boundaries can be difficult, especially considering that introns can be thousands of base pairs long. And so you have to really get a smart aligner, smart algorithms that can handle these things. And so that's why we use splice aligners such as STAR to be able to more accurately identify those non-contiguously aligned reads. And additionally, the STAR splice program is able to perform an unbiased search for splice junctions. So this is where it can identify novel splice junctions without needing any information on the location or sequence signals or the intron link. So it's going to be capable of aligning a read with multiple splice junctions. It's going to be able to align a read that might have indels and mismatches, and also it could be some with lower quality ends. So in order to improve the detection and quantification of splice sites, we're going to run STAR in that 2-pass mode. And again, that 2-pass mode is where the splice sites are going to be detected in the initial mapping to the reference. And then it's actually going to add information onto the reference that we already built in this step. So if it identifies, based on your data, novel splice sites that don't already exist in the reference that you build, it'll add those novel splice site locations to the reference. And then in the second pass, in the second mapping, it's actually going to be able to quantitate those novel splice sites. And so with this next command, we're going to align our trimmed forward and reverse reads of all 12 samples to STAR using the following command. STAR is the alignment step. It's going to require more computational resources than any other step in the pipeline that we have. So again, depending on the capability of your system, it could take anywhere from a few hours to even half a day or longer to run this mapping, this alignment. And so again, here are all of the options that I went over in detail in the lecture. So again, I won't go through all of these in detail again with you guys, but I do have here definitions for each and every option that we use in STAR. And of course, I always have links to the STAR manual. I think it's up here. Yeah, so here's a link to the STAR manual where you can find all the information for what all of these options mean. And the options that we use are all going to be listed here, and those are the same options that I went over in the lecture. In the input data that's going to be required is the STAR reference genome, so the output from the previous step. We need to tell it the directory that holds that STAR index. And then we're also going to have our FASTQ files, so our trimmed reads, which was the output from step 2A. And there's a bunch of output files that are generated from the STAR alignment command. So a detailed descriptions of all the STAR output files can be found in the STAR manual, which again is linked to here under the output file section. So the STAR manual is huge. It's like over 100 pages at least long, because again, it just has so much functionality. So if you just search that for output files, it has an index at the top, and you go right to the output files, and then it tells you detailed descriptions of all the output files that are generated. I'll go over the ones with you that are generated from our specific output. So we have a files that's going to end in a line sorted by coordinate outband. So this is going to be a binary version of the SAM file that we just went over. And that's an alignment map containing reads that map to the genome sorted by coordinate. Again, because one of the options that we used right here, this out SAM type, we told it that we want the output in BAM format. So again, a compressed SAM file. And we want them to sort the aligned reads based on the coordinate in which it aligns. Another file that we're going to get is aligned to transcriptome out BAM. So this is going to be another binary file, right? So another BAM file. And this is an alignment map containing sorting reads that are mapped to the transcriptome. Remember I told you we're going to get two SAM files, well, this compressed version of SAM files. So BAM files that are outputted. And this one is going to be the one that aligns to the transcriptome. It's actually the one that we're going to be using downstream. And that is generated because of this option here, which is prompt mode transcriptome SAM. So I'm telling it to also output a transcriptome aligned SAM file. We then have this log final out file, and that's going to be a log file containing the summary of mapping info and stats, such as the number of reads that were mapped, etc. So that's that file that I quickly went over with you guys in the lecture. Then there's a longer version of that file, so a log.out file, and this is like super long. It contains a lot of detailed information about the mapping run, and so basically if you need to troubleshoot, that's really the only time that you go and look at that file. And then there's also this log progress out file, and this is basically, will give you statistics as you go about how the mapping is going, so it updates you every minute during the mapping run. And then there's this Sj out tab. So this is going to be a tab delimited file, and that's going to contain your splice junction information and respective count data. So that's going to be the quantitation of the splice junctions that are identified, both unannotated and annotated. And then we have a couple directories that are outputted. So we have the star genome directory, and this is going to be the directory containing the extracted junctions from the first pass mapping that was inserted into the genome index. So remember, it goes through that first pass, and if it identifies any splice sites from your data that weren't annotated, that weren't already in your index, it'll add them to your index. And so it also gives you the files that were added to your index after that first pass. And then we have this other directory here called star pass 1, and this is going to contain splice junction and summary mapping and info from the first pass mapping. So this will be information that's only going to be your annotated splice sites. So these main output files, this Sj out tab here, that's going to contain both unannotated and annotated splice junction information. And then if you wanted just the ones that were annotated, you'd go into the subdirectory here, star pass 1, and it'll be the ones that were just from your initial index. And then there's this directory that's going to be empty. It's a temporary location that's developed to put temporary files that are generated as a program runs. And so with that, just before lunch, I want to submit this script. This script, again, is going to be a Slurm script because we're going to be running the same exact command on all 12 of our samples. And so we're going to do that by just submitting this Slurm array script, which will run all 12 samples using the same exact command, only changing the sample that's being run. So we can all go ahead and run that command, and you should see that you have submitted your job to the Slurm job scheduler, and it gives you a job ID. And so in the last minute here, while that's running, let's just take a look at the standard output, and then we'll answer the questions when we get back. So this is what the standard output looks like. Again, all this stuff up front here is the stuff that I add to the Slurm scripts just to tell me information about, remind me what job I'm running, the start time, the server that it's running on, and the job ID, as well as the sample name and the version of the tool. So this was run on star version 2.7.8a, and then it spits out the exact commands. This is all the options that we just went over. And you'll notice that the standard output just gives you this nice short summary, and it tells you when it started the run, loaded the genome, did your first pass mapping, finished the first pass, modified your index to add those novel splice sites, did your second pass mapping, finished the mapping, and then did the sorting of your BAM files to sort by coordinate. and then it should tell you that it finished successfully if it finished successfully. So when I look at these standard outputs, I am looking for it to tell me that it finished successfully, and I am making sure that there are no errors that were given out to me. And so you'll also note that this particular sample took about 2 hours to run, and it's a pretty beefy server that I put it on, so about 2 hours. With that, it is just after 11, so I'm going to have you guys all go and take your lunch, and when we get back, we're going to answer some questions about the standard output and then take a look at some of those BAM files. So if everyone could please be back here at noon Pacific time.